Well, good morning, ladies and gentlemen, and welcome to this webinar to give you an introduction to the exciting new Slimers project. My name's Tom Allen-Stevens. I lead the British On-Farm Innovation Network, and we also lead this project. And we're delighted to be working with UK Agritech Centre, Crop Health and Protection, or CHAP, with the Small Robot Company, with Harper Adams University, with Agravation, and with John Innes Centre on this project. Now, I'm just going to share my screen, if that's okay. Hopefully everyone can see that. So, strategies leading to improved management and enhanced resilience against slugs brings together two major research projects. Now, I'm going to leave it to our technical lead, Dr. Jenna Ross, to give you a full introduction to Slimers and how it really does promise a revolution in slug control. But first, I'd just like to update BOFIN members on some of our other activities and projects. So, many of you will have joined the Pulse PEP, the major new DEFRA-funded project led by PGRO, Nitrogen-Efficient Plants for Climate-Smart Arable Cropping Systems, or NCS. Now, you'll hear later about opportunities to receive funding for on-farm trials in Slimers, and we're doing the same for NCS, and there is a Pulses of Brilliance workshop taking place near Peterborough on 11th of September, where we'll be deciding with Pulse PEP members what activities we'll prioritise. So if you're not signed up already, please go to ncsproject.co.uk to find out full details on that project. And of course, Harvest is underway, and so is Harvest Weed Seed Control, our project looking at this exciting new window on chemical-free weed control of the trickiest of grass weeds. So Skye and I have been visiting our 4 weed seed destroyers who gave the Redicott, who have the Redicott seed control unit attached to the back of their combines to see how they're getting on. So do take a look on the BOFIN YouTube channel and do subscribe to get the updates. And thank you to all of those who have signed up as Seed Scouts. And I hope you've all received your scouting packs and are ready to take your pre-harvest sample if you haven't already done so. Now, thank you also to all of you who joined the Soil Squad and sent in root samples for analysis in our Soil Fungal Communities project we've been running with Crop Science Centre, kindly supported by KWS and RAGT Seeds. So at Groundswell this year, Dr. Tom Thurkle presented initial findings of our buscular mycorrhizal fungi in the roots that you submitted. Now, I understand he has all the full results from the analysis and will be sending everyone their results shortly. So look out for updates on this. And finally, our slug-resistant wheat project, year 2, is underway. And thank you to all of our slug scouts who gathered slugs and sent them in for feeding trials carried out at the John Innes Centre. We received nearly 1,000 slugs, and we have now got results back from initial tests on Watkins 788, the landrace wheat variety believed to be resistant to slugs, and on the Blackman line and a range of other modern wheats. So again, these results are summarized on the BOFIN YouTube channel, and we're just about to get underway with in-field trials on the 2 wheats looking to engage our slug sleuths, who I hope will also be taking part in Slimers. But we're also looking for another 1,000 slugs to continue the feeding trials. We have 84 recombinant inbred lines, so these are Watkins crossed with Paragon, to test which of these have inherited this curious trait. Well, apparent trait. We don't know whether it's a trait yet. So please get involved and sign up as a Slug Scout. We can send out packs with full instructions and also refresher packs for those seasoned heroes who have done so much for science already. So, but look, that's enough from me, and let's get on with the main event. And shortly, I will be handing over to Dr. Jenna Ross of CHAP to give you a full overview of Slimers. And then we'll pass to Professor Keith Walters of Harper Adams University, who will look at plans for the first-year trials and what our slug sleuths will be doing. I'll then take a look in more detail at what we're asking of the slug sleuths and how they will be paid to undertake their work. Then I'll be joined by Clive Backer of Aggravation and Yorkshire grower Graham Potter, who will talk about their experiences with on-farm trials. And then we'll open up for Q&A, and you'll see at the bottom of your screen a Q&A button and a chat button. So please use those to submit questions and to join the discussion. And we'll aim to answer as many of those questions as we can. And also, please use it for those comments as well. We do capture all of the comments that we receive in these webinars, and they really do help shape how we perform our activities and take these on-farm trials forward. So without further ado, hopefully, Jenna, would you like to join us? Would you like to turn on your video and join us if you're there?
Yeah, hopefully everyone can see me and they can hear me.
Brilliant.
Absolutely. Perfect. Thank you very much, Tom. I'm just going to see if I can share my screen now. Let me just do this. Okay, please let me know once I am sharing. Perfect. Excellent. Right, well, good morning, everyone, and welcome to the Slimers webinar. And thank you, Tom, for the introduction. My name is Jenna Ross, and I'm from the UK Agritech Centre Crop Health and Protection, or CHAP, as we are more commonly known. And I'm also the technical lead on this project, supporting Tom and the rest of the partners. But before I start speaking about Slimers, I thought it was important to give an overview of the work that we've done previously as part of an UK funded project called Slugbot, because it really helped set the foundation of where we're planning to take the Slimers project. So Slugbot, well, where did the idea come from? Well, back in 2017, I was working in South Africa on focused on the biological control of slugs. And I'd been overseas for about 10 years at this point. And during a trip home back to Scotland, I came across this headline in the Sun, some bit of classy reading here. 500 billion slugs set to invade Britain after springtime sex frenzy swells the population by 20%. Now, I always say there's two things that were surprising about this. First of all, that the Sun could combine sex frenzy, slug orgy, and slimy lust all into the same headline about slugs. But second of all, most importantly, is that I'd spent, at this point, 10 years living overseas. And it made me realize that nothing had really moved on when it came to slug control. And was there something we could be doing? And this really was the catalyst, the foundation for applying for Nuffield Farming Scholarship. So in 2017, I put an application in and was successful. And in 2018, I set off my travels. And I spent 26 weeks traveling the world looking at the future of slug control. And very quickly, I saw a gap in the market. And this was firstly how we can improve on slug monitoring, because right up until this point, it's quite laboursome. You've got to put your traps out, monitor your traps. And a lot of farmers weren't doing this. And it had led to an over-reliance on the likes of metaldehyde. And I also saw an opportunity when it came to precision treatment, especially around biological control, which is a particular passion area for me. So when I moved back to the UK in 2019, I took up a role at CHAP, and I wrote a bid, and this bid was known as Slugbot, which was subsequently funded by Innovate UK. And to give a proper overview of the project, I've got a very short video, and I'm hoping the video is going to work. So if you just give me a second, I will just pull my screen over, and hopefully this will all work for you. Slugs are major economic pests, both in agriculture and in horticulture. Current methods of control are reliant on chemical molluscicides, such as metaldehyde and ferric phosphate. There's also biomolluscicides available in the form of nematodes. However, they are not economical for use in arable and oilseed rape systems. And what we've seen is therefore an over-reliance on metaldehyde, and part of that is due to the lack of monitoring. And metaldehyde has massive impact both on the environment and also non-target organisms, and that has led to its subsequent ban. So therefore, this project is about developing an autonomous system to monitor slugs and understand where they are in the field, and then use that data for precision applications of biological molluscicides. The project is led by UK Agritech Centre, Crop Health and Protection, and working alongside robotic startup company, Small Robot Company, and also farming enterprise AAV and Enley.
SRC has two parts in this project, in the SlugBot project. The first part is taking all the data that Chap has produced and found for us of slugs, and using that to train an AI so we can actually identify two different sorts of slugs. And then the second part of the project is then using that artificial intelligence to be able to take action on a robot. And in this case, we're aiming to spray nematodes precisely onto the slugs in the first place.
I think farmers in general always look for new ideas to solve problems on farm, whether that's levels of production or less inputs and protecting the environment. And this project fits really well with that structure.
At the Agritech Centres, we work with leading scientists, farmers, advisors, innovators and businesses to understand industry challenges and their priorities, but also to develop innovative game-changing solutions for market adoption. And SlugBot is a great example. Here we're developing a solution that has potential to increase yields for farmers, but it also meets the ethos of sustainable, environmentally friendly farming with reduced chemical inputs. Okay, so hopefully that helps give a bit of an introduction into what SlugBot was about. Bit more information. So SlugBot was pretty much broken down into 3 phases. It was about collecting slugs, and we did that right across the country, and that was our project kicked off during lockdown. So we'd be quite innovative in how we approached that. So we took very much a citizen science approach. It involved multispectral imaging and RGB imaging, so normal photographs of those slugs, and all of that fed into an artificial intelligence model. At the same time, we were trialing the slug mapping capabilities, and we did that in the glasshouse and in the field. And then phase 3 was all about developing that precision spraying capability that was mentioned in the video. We looked at 2 slug species, the Arion vulgaris, which is the big Spanish slug that some of you might have seen, especially in the southern half of the UK, and the Drosophilus reticulatum, the greyfield slug. And we chose these 2 because they are particularly pestiferous. We then imaged those slugs in the lab using this bit of equipment on the left-hand side called the Videometer. And we imaged them, both the slugs as well as looking at background material. And again, all of that data fed into the AI model. We also tried to support that data with molecular work and also the morphological work. So that involved dissecting slugs just to confirm the species that we were dealing with, especially those Spanish slugs, because they can be quite tricky to confirm identity. We then gathered continual image data, and that was done both in the glasshouse, as you heard in the video, as well as doing field imaging. And all of those images, as I said, fed into that AI model, which was then adapted in the robot for monitoring and geolocation. And that final piece of the puzzle, as I said, was about developing that spraying capability. And the last iteration for our project, we were able to spray down to a 20 centimetre squared area, controlled by the microcontroller, pressure regulated, flow measured, and also calibrated. And a project like SlugBot can only happen because of a team. It's not just one person, it is a multidisciplinary approach. And what excites me about Slimers is it really is the dream team. We're bringing together the team that was involved with SlugBot along with the team that was involved with project at likes of Harper Adams University, and we've got the likes of John Innes Centre, bringing us on board Aggravation, and all led by BOFIN and obviously funded through RUK. So what is Slimers? So strategies leading to improved management and enhanced resilience against slugs. Not quite as snappy, so hopefully Slimers is a little bit easier to remember. And ultimately, what we're trying to do is build on previous work. So build on that work that we've done with SlugBot, and build on the work that Keith at Harper Adams been doing, and he'll be speaking about that shortly. And bringing together this amazing expertise in a consortium. And what we're looking to do is develop a cost-effective digital autonomous slug monitoring and spot treatment system. We want to develop a slug forecasting and precision application tool. And ultimately, we're looking to develop that knowledge and understanding so that we can control slugs, but do that in a sustainable way. And ultimately, what we want to do is reliably reduce that reliance on slug pellet usage by advancing these alternative biological control measures, such as the use of the nematodes, but do that in an economically viable approach. So we have various outputs that will come out of this 3-year project. We'll be looking for the actual tangible physical digital slug monitoring, forecast and precision tools, and they will be delivered by the end of the project, aiming for an end-to-end service model. So really taking a lot of the R&D that we've done from the feasibility work of SlugBot and other projects and taking that further towards commercialization, and that will be supported by the commercialization strategy. There's also huge amounts of unknown when it comes to slugs, so we'll also be looking to gather data on things like slug behavior, distribution, surface activity, feeding, biology, even right down to species diversity. There's a huge amount of information we can gather as part of this project. We'll obviously be working with our slug sleuths, so it really is about bringing research, farmer networks all together under one roof, and we're desperately always looking for farmer feedback. So that'll be about gathering data from your farms, but also supporting that with market research. We've got various different dissemination events taking place throughout the project, and Tom will be keeping all of you up to date on those in the coming months. And this will all be supported by various literature materials, you know, the social media platform, as well as knowledge exchange platforms. And again, Tom will be giving you updates on all of these. So how does the project look? Well, ultimately, the project is split into 4 major work packages. So work package number 1 really helped build on that work that was done in SlugBot. So we will continue gathering that user assessment research, and that's really about ground truthing and understanding what's going on the farm, what the demand and need is. We will continue gathering that imaging data, and this is so important, especially when you want to develop an AI model. We need something that is robust, but not overly trained. So we need to give it really diverse data sets from as many different farm systems, as many different background materials as possible. And we'll be doing that both through manual data collection and then moving into robotic data collection later in the project. We'll then be doing the updated iteration of the robot, which will be done through Small Robot Company. And again, as I say, updating these AI models based on the data that's gathered. This will then be layered along with other data, which you'll be hearing from in more detail for Work Package 2 shortly. And these will all be bringing together along with that hotspot modeling. And then finally, it's about continuing to develop that treatment capability. So I mentioned about the spot treatments. We want to take it further than we've taken it, 1 step further. And that will be the final plan of the project, which will leave us with our final demo of a prototype. Now, Work Package 2, you're going to be hearing more about this shortly, but a very brief overview. It's about looking at the prediction of patch location. So we'll be building on the earlier work done through Harper Adams University and others by challenging and confirming the hypothesis that selected factors, be that sand, clay, silt, organic matter, percentage content, are all sufficiently strongly related to slug patch location to be used in patch treatment methods. We'll also look at the cost-effective method for patch location prediction. So as the selected factors all affect soil moisture retention, we'll be trialling the reuse of soil mapping data, and you'll be hearing more about that shortly. And really, we're doing this to test their use as proxies for soil sampling analysis in a commercially viable system. And finally, it's about looking at the efficacy of control achieved by patch treatment. So we'll be comparing patch treatments versus a full-scale field treatment, and we'll be testing the hypothesis that patch treatments will hopefully significantly reduce the amount of molluscicides that will be required to maintain slug populations in order to allow us to achieve that AHDB damage treatment threshold levels. And then finally, Work Package 3 is all about farmer engagement and dissemination, which is going to be led by Boffin and by Tom. And here it's all about building up, obviously, our slug sleuths, which is all about today. It's about developing that slug circle and bringing various stakeholders together, and then feeding that into the wider innovation community. And this is all going to be supported, as I mentioned earlier, by various dissemination materials, be that written, webinars, live events. And again, you'll be hearing more from Tom, not only today, but as the project progresses. So I'm going to finish there, but my contact details are on the screen, so please do get in touch or in contact if you're keen to learn more about Slimers and also CHAP's wider work. And I believe I'm now going to pass to Keith, who's going to tell you a little bit more about their work. So yeah, thank you very much for your attention, and please do enjoy the rest of the webinar.
Great, thank you very much for that, Jenna. That's a super introduction to this project. I have to say, it's fantastic to work with people like Jenna and other people on the project as well. It as she says, it's a really great team. Now, please do thank you very much for your questions and comments and so on. Please do keep those coming in with the Q&A at the bottom. We're not going to stop for questions now. We're going to move straight on to Keith. Would you like to switch on your video, Keith? And I think I'm running your thingy, so let me just your slides. So let me just get that going. Hopefully that's going to work. Yeah, are you there, Keith?
Yep, indeed. Can you hear me?
Fantastic. Yes, we can.
Okay. Well, thanks for that, Tom. Thanks for the introduction there, Jenna. What I'm going to do today is give you just one slide of background for WP2, the work package which Jenna was talking about, and then go straight on to a couple of slides indicating what we may be asking slug sleuths to do this year in the first year of the work in the field. The work we are going to be doing builds upon about 5 or 6 years of work at Harper Adams University, which we've been doing in conjunction with industry in order to look at the sustainable use of slug pellets. Now, that may appear slightly different to what Jenna has said, but Tom recognized when he heard about this that actually the same work could be used and duplicated in order to help tweak the economics of the slug bot nematode control towards something which is commercially viable. It's built on a simple premise. Slugs, as you know, across the field, an arable field, are not evenly distributed, but live as patches in which there were of higher densities of slugs in a sea of lower slug numbers. The important thing that we've shown is that these patches are stable in time over a whole season, probably between seasons. They always occur in the same place. And this gives us the opportunity to say, okay, initially, can we target the application of ferric phosphate pellets at these slugs while still maintaining effective control? The answer to that has been yes. We now know not only that these patches are stable in time and space, we know also why. Behaviorally, we've worked that out and it's really well established. And so we can, with modern equipment, talking to the equipment manufacturer, bolt on a geolocation equipment onto the back of the existing pelleters and apply those pellets accurately. We've also done some work and we've shown that actually the initial work shows that by targeting these patches, we maintain commercially effective control of the slugs in the same way as you would with a blanket treatment across the whole field. And so everything looks good. But, and there's always a but in these, what we need to do, as Jenna was saying, was accurately predict the location of the fields in advance of the start of the season. If we don't do that, then obviously we cannot trap with slugs, slug traps to find out where they are, and the whole thing is economically not viable. Again, as Jenna was saying, we've done some initial work on this, and we found that slug patch location is correlated to specific soil characteristics. There's a number of these, but in particular, the percentage of sand, silt, and clay in the soil is important. The organic matter content of the soil is important, and to some extent, pH is important. Other factors contribute, but those are the dominant ones. Now, if this relationship is sufficiently strong, the relationship between these factors and the location of the higher density slug patches is sufficiently strong, then we can quite easily, we think, then target the applications predict where the location of the patches is going to be in advance and set up the equipment so when you apply pesticides, you will target the applications bang on these patches. And so the objective of year 1 of our work is quite simple. We need to establish whether the relationship between patch location and soil characteristics is indeed strong enough to be used. Can I have the next slide, please? Thanks, Tom. Okay, now the work which we plan for year one involves 4 basic steps, a subset of which we'll ask slug sleuths to do, some of which we'll do ourselves. The first thing is to record the important field and crop details and input details. We would ask people to provide once before the beginning of the experiment, and then that will allow us to look at the normal farming husbandry activities which might be applied to a crop and see if just check that that doesn't affect the location of patches. Our data so far suggests it doesn't, but we need to be certain. The second one is to lay out a large field trial plot. That's illustrated in the little diagram to the right of this slide. It's a 1 hectare plot, 1 per field. It measures 100 meters by 100 meters, and in the middle has a grid of traps marked by the axes on that diagram, with separated with 10 meters to the nearest neighbor. The traps are quite simple things. They're upturned flower pots. We might describe them differently to make it sound good, but that's what they are in reality. And they're just laid with a stone on the top to stop them being blown away. That's fine. Why 10 meters by 10 meters? That's actually the result of a lot of modeling and field experiment work, which we've done already. The problem is that if you have patches, as we do have, which are relatively small, then if you have those traps placed too far apart, then as illustrated by the little blue blob on the left of the diagram, they fall between traps and you can miss the patch, and we don't need to miss patches. On the other hand, if they're too big, as illustrated by the larger blue blob on the right of the diagram, then you will find several traps occur within that blob or that patch, and you're wasting sampling effort, you're wasting people's time by taking too much data. So the 10 meters is fairly critical and isn't just guesswork. 10 meters between traps. Okay, so that's the trial layout plot. The third thing then is to go out and actually use the grid and count the number of slugs under each trap on 5 occasions after crop emergence, about 50% crop emergence. We do offer, for the enthusiastic slug sleuth, the opportunity to take further counts if they wish to, perhaps 3 counts at approximately monthly intervals thereafter. That would be absolutely great, but the critical thing is we must have those 5 initial counts. And step 4 is to take a single set of soil samples and send them into the laboratory for analysis. We can then look at the variation of the soil characteristics we're interested in across this 1 hectare trial plot. We can compare them mathematically with the distribution of slug numbers across that trap, and we can do some mathematics just to make sure that we know how strong the relationship between the two is. Okay, Tom, could I have the next slide, please? Okay, so that's the overview. In detail, what will we be asking slug sleuths to do, and what will we do ourselves? Well, clearly the first step, the recording of field and input details, are for the slug sleuths. You know what you're doing, so we will ask you to fill in a form we will give you in order to record that. The form will be made available either on an app for your mobile, or for those of you who prefer on paper, but those details are very important. Step 2, the laying out of this, on the face of it, rather large and complicated trial, is something which we will do. Well, I say we, that's the kind of the royal we. Clive has volunteered to do this, and thank goodness he does. He is extremely good at this. So other than agreeing where in the field that trial can lay, and agreeing to us to set it up, you have to do, as a slug sleuth, nothing more. Step 3 is something which we would ask the slug sleuths to do, and that's take the account of the slugs that are under the trap. As I said, there's from about 50% crop emergent, we would like a 5 assessments made at weekly intervals. And again, we will give you a form to record those details, which will be available again as a paper copy or preferably on your phone as an app. It is very important that the counts on individual slugs are associated with the exact correct trap in the grid. That's easily done, and the design of the app guides you through that, so you don't have risk making mistakes. And the other thing we would like you to do is take 10 or 20 photos at some trap locations using your mobile phone. And that's so that we can check that the damage done by slugs actually does relate to where the slug patches are and where there's lower number of slugs. Again, that's a belt and braces check to make sure that we're not presenting any risks with patch treatment. Tom, could I have the next slide, please? Okay, so when you're counting the slugs, what do we want you to record? Well, there's a lot. I think there's 51 species of slug in the country at the moment. Of those, the grey field slug is the most important pest generally in the crops we're looking at. And so we would like you to record the number of grey field slugs under each trap. And in addition, and separately, we'd like you to record the total number of all other slug species under each trap. We don't ask you to identify them to species, it's just the total number. So if you could give us 2 figures for each trap, one is the number of grey field slugs you look at, and one is the total number of other species. That's all we need. And that's simply it. So that's step 3. And finally, step 4, at the second visit, we asked to take up to about 100 grams of soil at sample locations next to each of the trap. Now, again, we don't ask slug sleuths to do this. That would take a little too much time. It would not really, we felt, to be fair. And therefore, Clive Blacker has again agreed to volunteer to do this. Final thing I would mention is that if you have an EMI scan or Omnia Terra map, then in order to automate this trap location approach, then we would ask you to let us have access, if possible, with that. If you haven't got one, don't worry. Clive, again, in most cases, will be able to provide a free scan, an EMI scan for you. Why do we want this? Well, a lot of the factors that we're interested in terms of soil characteristics, which will promote or identify the location of slug patches, rely on the retention of moisture. And these scans are very, very good at indicating areas of the soil with high moisture retention capabilities and lower moisture retention capabilities. So the ultimate idea, which we will be testing in years 2 and 3, is to reuse such maps when they're taken for other husbandry purposes on the farm to predict where the locations of these higher density slug patches are, and to apply and use that as a basis for applying the pesticides. So again, these scans will be extremely useful throughout the project as we're building a data set to test and optimize the system. Tom, I think that's all I have to say. Does that cover it?
Great. No, thank you very much for that, Keith. And as I say, we'll move straight on, but do please keep those questions coming in, please. And if you've got any comments to make as well, please do use the chat room. I'm going to just give you a few more details of what we're expecting of the slug sleuths. Now, it's important to understand here that this is it's funded by Innovate, this project, and one of the core elements of this is that the slug sleuths will be paid to do this work. And this is really quite a new aspect of this sort of research work. We're going to be paying you. That's going to be, the contract will be with BOFIN, so you won't have to deal with Innovate UK. And I can tell you that if anyone's dealt with Innovate UK, you will know that it makes RPA look like a walk in the park. So the idea, the way that the contracts will be structured is that you will receive a contract fee of £2,500 for doing the work. But we charge you a membership fee of 30% for all the admin and all the various resources and so on to do the work. So there's a net fee available there of £1,750. The way that's paid is that you will send me an invoice towards the end of November, and we'll pay the first half of the invoice by the end of November. And then the second half of the invoice we will pay by the end of May. Now, there are 3 roles that you have to perform, and Keith has explained the roles of trial host and trial monitor very adequately, so I won't go into too much detail there, but there is another role, which is project ambassador. And again, this is a very new role within this sort of research work, and it's important, really, really important about sharing the information within this trial and sharing your knowledge as well. And I'm going to repeat this phrase time and time again over the course of the project. Knowledge has value. Your knowledge has more value than any other knowledge in the industry. And that's what we're looking to unlock within this project and really make it valuable for everyone to share within this project. So I'm going to say a little bit more about that now. Bear with me. So as far as the support that we're going to be giving for Slug Sleuth, and Keith has already alluded to this, we'll provide you all of the materials you need in terms of the slug traps, the white canes, and so on. There'll also be a Slug Sleuth handbook, which gives you chapter and verse on how to run the trial. And as Keith pointed out, aggravation, Clive Blacker will be doing quite a lot of the, they'll be doing the setting out, and they'll be doing the soil sampling as well. And we're talking to Clive in a minute. He'll give a few more details about that. There is an app, and we very much encourage you to use the app. It captures all the information together, and it also brings the photos to the trap locations as well. And that is by far the best way to submit the data. And there's also a WhatsApp group, and obviously we'll give phone support as well. And you'll be hearing from myself, you'll be hearing from Sky, our community manager, as well. And we'll obviously make sure that you have everything you need to perform this trial. So let me just tell you a little bit more about being a project ambassador. So here is a list of the things that we'll be asking you to do as a Slug Sleuth. And the ones in white, Keith's already talked about the elements of setting up the trial, of monitoring the trial, and of delivering the data. Those ones that are shaded are to do with involvement in the trial and things like attending this webinar. And you can accrue points over the year for your involvement within this as a Slug Sleuth. And you'll need to accrue 40 points in order to get the second half of your contract fee. Now, the 40 points sounds like a lot, but there is going to be a workshop in March where you can accrue 18 points for that. Every time you turn up to one of our events, and we're planning to have at least 2 over the next few months, then you can accrue 6 points. If you're involved in a press feature, and we're looking for lots of involvement with the press and so on, if we involve you in one of those, that's another 3 points. And also there is the Slug Circle, which I'm going to tell you a bit more about in a minute. And every contribution on that is worth a point. And it doesn't take very long to accrue quite a lot of points on this forum, which I'm going to tell you about in a minute. Now, the important thing about this is that we want to encourage and support you through this. We're not the RPA. We are not Innovate. It is taxpayers' money. We have to ensure that the taxpayer gets value for money, but we're not here to police it. We're here to support you. We're not here to penalize you. We're here to make sure that you accrue all the points that you can and to get your full contract fee. And that's a really important element of this. Now, I want to tell you about the slug slurp. This is entirely new. So this is a new area on the Farming Forum. It's currently hidden, but we've just been building it, and it's now all ready to go. Sky has put some information on there and resources. So it's a place, it's a central knowledge hub where we can go, and we're going to be putting documents on there and interesting elements to do with slug control, to do with patch location and so on. We're going to be putting on there. But it's also an area of discussion, and this is where your knowledge really comes to the fore. We want your involvement with this. It happens, I know you do it on Twitter, you do it on the Farming Forum and other places. You have these brilliant conversations about what you're doing in the field. We want to bring them all into one place because that knowledge has value. Now, the idea of this forum is that every contribution that you make, every contribution of value, if it gets a like, then it uprates it. And that makes it easier to find, easier for other people to find. And then you can then rate the comments on there, and you can see which ones are getting the most likes, if you like, and which are getting the most value. And we can then, so the more things that you raise on there and the more people like them, the higher your rating gets. And that is how we're looking to value this site. And over time, I hope that this is going to be a knowledge resource that will actually, that those members who contribute most will actually be able to make money from their contributions. But more on that later as the project develops. But that's why I say knowledge has value, and that's a really important part of this project. So this platform will be going live shortly, and you'll be invited to come and join it. And obviously, the earlier you join it, the more contributions that you make, the more points you make if you're a slug sleuth, if you sign up as a slug sleuth. I'm going to, with time's rattling on, and I'm going to pass on now to, well, I'm going to invite, if you could, Clive and Graham to join us. Are you there, Clive and Graham? I'm going to stop sharing my screen. Clive, hello. And do we have Graham there as well? Do you want to turn on your video? Brilliant, fantastic. Clive, would you like to just introduce yourself for everyone?
Yeah, so I'm Clive Blacker. I run a small business called Agrivation, and my background is precision ag and agriculture. So I come from a family farm in the Vale of York, not a million miles away from Graham, and we have really variable soil types here. And I started with precision ag, getting into it really through trying to manage our soils better and more profitably with precision technology, and have been doing so ever since, and now help others to do so.
Fantastic. And Graham, would you like to introduce yourself?
Yeah, I'm Graham Potter, up in North Yorkshire, yet again, not too far from Clive. We are a 500-acre arable farm, and we also do contracting as well. And I was a slug sleuther last year, and quite enjoyed doing the trials. So, yeah. Brilliant.
Yes, no, that was a slug sleuth in the slug-resistant wheat project, I think, wasn't it?
Yep.
Yep. So, as I said, I talked about that a little earlier on. Do look out for updates on that. But now, turning to you first of all, Clive, we heard from Keith, and I've also intonated as well, that there's a certain amount of work that you're going to be undertaking of the trials. Would you just like to say a little bit more about that?
Yeah, so I'm committing to make sure that we collect the certainly baseline data in an accurate fashion. And take all that hard work away from the farmers. So the work that you guys are going to be doing on farm can all be laid out. It's all set out. All you've got to do is identify and count the slugs. So what it will involve is I'll come and conductivity map your fields if you haven't had it done already. Although there's a great chance I could probably do that again for you and just make sure it's relevant to what we're mapping with. So we're using consistent data across all the sites. Ideally, we'd like to do that as soon as possible, so we can get that information and understand the variability of each site before we can then put the traps out. So it'll give us a good indication to discuss with you guys the soil texture maps that we can produce from that data, to identify where the variability is, to correlate that with your slug patches. So when we put out our hectare block, that we've got it in the most representative area that can show soil textural change, but equally be a really important place for mapping the slugs.
And then you'll be helping set out the traps themselves, won't you?
That's right. Yeah. So then once the crop's established, so you guys will then drill the fields. You'll plant your crops, and once it's drilled, then I'll come out and I'll lay out the hectare plot in the field. I'll put the 100 points out and take the soil samples at that point, potentially as well. And there's a good chance that I would like to try and at some point also map the fields with drones as well, just to make sure we've got a good variable understanding of the canopy as it emerges as well.
Yeah, and then Keith talked about the soil sampling. That's another aspect, I think.
Yeah, so we'll be taking a soil sample core from each of the upturned or each of our slug traps, so that we have a really accurate correlation to the soil organic matter, particularly content, I think, and the soil texture content that Keith will then do. So yeah, taking 100 soil samples from each site, and then delivering those to Keith so he can then do the processing.
Great, fantastic. Well, turning to you, Graham. Now you've done quite a bit of research work on your farm previously. I know you have, but I mean, this trial is asking you to look at, monitor 100 traps. Are you ready for this? How do you feel about that?
A little nervous. When we were monitoring the traps last year, yeah, it was, it took about maybe 20 minutes out of your day to go and do it. It wasn't a big job. The app worked fairly well. This time, monitoring 100 traps, I'm hoping the app is very, very easy. It'd be easy if we could just take a picture and go to the next one, take a picture. When you start filling in data on the app as well, it all takes time. So yeah, doing 100 traps could be taking an hour or more out of our day, especially during a busy period. But yeah.
Well, as you know, Graham, so we introduced the app last year, and you very kindly helped us shape it and use it, and hopefully it is more usable this year and better. I mean, it is great in terms of bringing all the data together. Isn't it? But I think you're probably right. About an hour for to monitor the.
My thoughts were, I know you were saying taking odd photos, but what would be a lot easier to do, and probably for Keith as well, is if we just lift the trap, pick it up, take a picture. He'll see what species of slugs are on there, and everything's all there in the picture. Then you guys can count them yourselves. I mean, that would be so quick to do that.
Although I'm not sure how Keith would feel about receiving 3,000 photos to have to count slugs on once a week. But perhaps we can develop an AI model or something that will do it automatically.
That would be great, wouldn't it? But no, I mean, yeah, if it's dead simple and we just lift up, put a number in, and that's all we have to do, then that'll be fine, I think.
If it helps, 6 years of experience has completely been wasted, because Graham, you've estimated the time it takes perfectly. It is about an hour. It gets quicker as well as you're practiced. Sounds strange, but it does. Yes.
There we go.
Good.
No, so now you've Graham, you've been involved in research on your farm for quite some time. Do you want to just give us a little bit of an insight into the sort of things you've been doing?
Yeah, so I've been a direct driller for quite a long time. We were on the Claydon system, so we were realizing we were having quite a few problems with slugs once we went away from the plough and everything else. So I brought it on myself to start doing a little bit of research with slugs, because there was nothing out there there was, Byron going putting slug pellets on. So back, I can't remember when I did it now. So we straight after a field of oilseed rape, we just went and put some wheat straight into the field out of the shed. and we started. We were experimenting with double rolling, rolling at 90 degrees. That we drilled it, and we were trying the difference between using metaldehyde and ferric at the time.
Yeah.
Yeah, so sorry about that.
And I think you.
So we did it in blocks, and then we would go along and we'd go and look and monitor and see how everything had been tubed. So from these little experiments we did on the farm, we learnt that on certain soil types we should double roll. And then another thing with the conductivity maps, we also know where slugs like to hang out more, and where they don't. So we would go and we actually are applying all our slug pellets at the moment we have been for a long time, invariably. So on some of the heavier soils where the slugs like to live, where it's a little bit knottier, they'll get a bit more of a dose. Whereas on the lighter areas, on the lighter soils where we don't tend to have the bother, then we don't apply any pellets at all.
And tell me I mean, you've heard from Keith and Clive and so on about the patch location and how we're going to be, sort of trying to get that a little bit more precise. Well, a lot more precise, I would hope. How do you feel that? I mean, is this a service that you think would be useful for you?
Yeah, definitely. I mean, what this will do, it'll actually, if my theory is right, that slugs like different soil types, and where I intend to put this plot, hopefully, it will say, well, yeah, actually, we are right, or they do actually just like to live in anything. It doesn't matter what it is, but so yeah, if we've got this hectare plot between 2 or 3 different soil types in one field, which we can do, I'm hoping. I think we'll be learning a lot from this because, hopefully my theory will be proven that slugs don't like the lighter soils. They prefer the more of the medium loams. Actually, some of the really heavier soils, they don't tend to bother much either. So I know there's one soil type in our farm that they seem to thrive in. So yeah, learn a lot from that.
I guess you'll get a clearer idea of that. Clive, turning to you. I mean, you've been involved in precision farming for many years. I mean, this is what lies at the heart of this project. How do you, as someone who's seen these systems evolve, how do you see precision slug application evolving through this project?
I think there's lots of opportunities. I mean, the biggest limitation we have is the pellets and access to pellets that we've got. So what we've got to use more responsibly and be more proactive about it. And slug control, if we can be proactive, can be done very effectively. And as novel treatments emerge, the work that Jenna's been doing, certainly with the nematodes, is really exciting. But to get it effective, we've got to be able to treat the target effectively, and just the areas of it because of the cost of it. So precision really plays into that opportunity to deliver things effectively in smaller areas.
Yeah, well, on that note, Jenna, is there any chance you could join us if you could switch your video on and your audio, and Keith as well, if you'd like to join us. Are you? Do we have Jenna there? I was hoping to bring in Jenna. Well, listen, while we're waiting for Jenna to join us, Keith, bearing in mind what Clive's just been saying about this patch location work, I mean, I don't know whether you've made any attempts in the past to actually sort of bring this into commercial practice. How do you see such a service panning out?
Right. It's interesting in that there's a lot of steps in order to take these things through to something that's commercially viable, all of which really need to be done in close conjunction and driven by industry. We've been doing some of this already. It's interesting that, not surprisingly, somebody who knows their land, like Graham, has come up with exactly the points which we're trying to test and prove, which is encouraging. What we need to do is make sure that whatever we have is robust enough to allow farmers to retain complete control over every aspect of crop growing. So we don't say, if you're using this system, don't do this. We need to know that irrespective of what you need to do to grow your crop, the system works. We need to know also that the equipment and all the costs associated with it are no more than those which are associated with a blanket treatment. And that's certainly we've looked at, that is the case. We believe, talking to a lot of equipment manufacturers, that standard equipment can be easily used by bolting on, for example, standard geolocators, so that you can then predict where the patch is, and just everything is done automatically. So we've done a lot over the 6 years. We've worked right from the beginning with people like Clive, manufacturers and what have you, to make sure that the way the work has been done allows and supports this ongoing assessment of, is this viable?
Yeah.
Interesting. I noticed in the chat earlier, somebody asked the question, for example, to the hectare plots, do we keep them free of pellets? Do we apply pellets?
I was going to ask about that.
Yeah, and the answer is, we want you to use exactly normal farm practice. Whatever you do to the rest of the field, please do it to the plot. And it's simply because we need to know that if you apply pellets, you're not actually removing these higher density slug patches.
Keith, just before you carry on, when we do start doing this trial, now we always pre-bait before we drill.
Yep.
So we're actually going to get rid of quite a few slugs before we drill. Now, obviously, these plots are going to be put in after, so we really don't want to be applying any pellets to this plot before we drill, like we would with the rest of the field. Otherwise, when you want to start, we might have had a really good kill and there might not be any slugs kicking about, so which kind of defeats the object.
Yes, but there is the issue, of course, that at any one time, the majority of the slugs are below the surface of the soil. They only come up to the surface occasionally. And that is magnified when you drill because the food sources are down there. They'll be going for wheat seed. And so even greater proportion of the slugs will be under the surface.
But don't slugs always come up to the surface to graze on the night time, don't they? So they come up to the top first to look. That's why we pre-bait before we drill.
Yeah, they do, but not all of them come up every day, every night. They come up and down. That's the beauty of the slug pellets, in that you lay a trap for them. When they come up, you've got a nice tasty slug pellet there to bait. Now we
basically we just do what we normally do then, and you don't mind.
Do what you normally do. And what we need to know is whether the patches stay where they are, irrespective of how you treat them. And so far, the work has a strong indication they do.
And no, I was going to thank you very much for joining us, Jenna. I was going to turn to you, just ask you this nematode treatment that you talked about, that you looked at in Slugpot. Can you really see that coming out as a commercial service at the end of this?
Yeah, no, absolutely. But I think the key to that is making sure that we can get the economics to work, because nematodes are an expensive alternative. But yet they have a significantly less impact on the environment. They are ultimately a natural parasite of slugs and snails anyway. But for us, it's all about getting the economics to work. We need to make sure that when we're spot treating, we can get it down to a small enough area that the cost isn't more significant than using pellets, but also making sure that we don't get it so small that the cost of your spraying perspective becomes too expensive. So there's a real balance to get those economics working.
I was just going
to say, Tom, but if we do get it to work, it becomes really exciting, because in this case, we're speaking about slugs, but ultimately spot treating with biologicals, whether they are micro or macro, like nematodes, if we can get that to work from an economic perspective, it opens up a whole new opportunity, not just to slugs, but to other pests, diseases, and weeds.
They're fantastic. Gosh, listen, I'm so sorry. We've run out of time. Thank you very much for the comments and the questions that you've put through. I think we've covered most of the ones that have been asked directly as we've been talking. I've been keeping an eye on things, but we will gather those comments. If there's anything that hasn't been addressed, we will make sure that the person who addressed it gets a response on that. But as I said, we've run out of time. The next steps, by the way, we are currently recruiting for Slug Sleuths. We've already got in touch with many of you who have already expressed an interest in being a Slug Sleuth. Let me just quickly share my screen and I will bring up my contact details. Bear with me. Whoops, that's the wrong one. Hang on. There we go. So there are the contact details for myself and Sky. Please get in touch with us. If you're interested in being a slug sleuth, we will send you the heads of terms of the contract and the slug handbook as well, the slug sleuth handbook to have a look at. We are looking to bring people on board and to actually sign them up by the end of this month, preferably. Certainly before drilling starts, obviously. And we are really keen for you to be involved. And I'm more than happy to talk through the actual terms of the contract with you and what's involved with anyone who wants to be involved. So, well, listen, thank you very much, everybody, for joining us. Thank you especially to Jenna, to Keith, to Clive and to Graham for joining. I have to say, I'm really, really honoured and privileged and excited to be heading up this project, not just because it's a fantastic team of people to be working with, but also working with yourselves, you know, some of the most progressive growers in the country, and actually doing something that is really going to make a difference to slug control and make a difference to how we manage our farms. I mean, remember that the only form of slug control we currently have are these ferric phosphate pellets. That's not a great and resilient way to be at the moment. And what we're going to be doing is going to be potentially extremely exciting. So on that note, thank you very much for joining us and have a fantastic day. Hopefully there'll be some harvesting going on today. And do get involved. Let us know more and we will keep you informed about how to join the Slug Circle and about how to stay involved in this project. So thank you very much, everybody, and goodbye.